Well, thank you for the invitation. This presentation is going to be a little bit different. I'm a pediatric nutritionist, and I study the impact of milk components on the gut and also the microbiome. So what I'd like to do, and I'll go briefly over this because you've heard a lot about the microbiome gut-brain axis, talk about human milk and effects on cognitive outcomes, some of the work we've been doing recently using the piglet as a model, and then provide some conclusions. So as you've heard already, the gut-brain axis is this bidirectional neurohumoral communication. And it's quite complex, including hormones, cytokines, metabolites, and more recently, we've appreciated the role of the microbiome, mainly through studies in germ-free animals initially, and then also from clinical studies. The point I want to make about the infant is that they're born developmentally mature with a very low level of colonization. There's some controversy whether they're actually sterile at birth, but either way, that if they are colonized, it's at a very low level. And studies have shown that this period of time is critically important for programming lifelong health. So this is a figure from the group here in Cork, and it basically shows the early life events affecting the microbiome. So we have mode of nutrition, breastfeeding versus formula, mode of delivery, cesarean versus vaginal, genetics, hospitalization, antibiotics, environment. So for example, preterm infants or cesarean-derived infants tend to be colonized more by environmental microbes or skin microbes. Also maternal infection, disease, stress, and the gestational age. One thing I'm going to talk about today is the bioactive components in human milk, which go beyond some of the prebiotics that are routinely added to infant formulas. So this is some data from my laboratory looking at the fecal microbiota of 3-month-old infants that were either exclusively breastfed or exclusively formula fed. And this is both at the phyla and the genera. And one of the striking things that we observed in this paper was this orange is the Bacteroidetes. And when we looked at the formula-fed versus the breastfed, so this is the Bacteroidetes to firmicutes ratio. And other studies have shown this. I think it depends on the formula it's being fed. Some studies have shown that there's Bacteroidetes, but it interests us because there was, work in regards to human obesity, which showed that this firmicutes to Bacteroidetes ratio was very important. So we're looking at this as a related to childhood obesity and early programming. And then if we look at the general level, again, you can see the formula-fed infant is actually much more diverse. And we see about the same amount of bifidobacterium, but as was previously mentioned, once you go to the strain level, you see big differences. So the breastfed infant primarily has B. longum infantis, which was something that was studied in the previous adult subjects, whereas these have different strains of bifidobacteria. So if we look at the postnatal, we do have windows of opportunity for influencing the microbiota and the gut-brain axis. So this could be supplementation to the mother, which would impact the breast milk, but it could also be supplementation directly to the infant through pre-probiotics or other components. So talk a little bit about breast milk and breastfeeding in human milk. And we know that human milk is the gold standard for infants. It provides all the nutrients necessary for normal growth and development and reduces the risk of many diseases, infectious diseases early in life, but also programming of health later in life and reducing a number of immune-associated diseases, even things like type 1 diabetes, IBS, and other autoimmune diseases. But the thing to keep in mind is that breastfeeding is more than just nutrients. There's social and tactile interactions between the mother and the infant. And where my lab spends a lot of time is in this area of bioactive components. And so the other thing to keep in mind, and as we think about early nutrition, whether it's breast milk or infant formula, it's impacting the establishment of the microbiome, as I showed you. It's impacting intestinal development, which has been clearly shown in animal models. And it's also impacting immune development, both mucosal and systemic. But then all of these are interacting with each other. we know from germ-free animals, for example, that in the absence of a microbiome, the intestine and the immune system do not develop normally. And all of these things, particularly barrier function, are implicated in the microbiome gut-brain axis. So how might milk bioactives influence it? There could be direct effects on neural development. This has been shown for some of the lipid components as well as proteins, and I'll talk a little bit more about that. Influences mucosal and systemic immunity, shape the gut microbiota as well as the metabolites, and modulate intestinal development and barrier function. So there can be specific effects to the gut, specific effects in the brain, and then we can see the interaction between the two. So we've been focusing a lot on cognitive development, although we are looking at other aspects of neurodevelopment as well. And this is a summary of a meta-analysis that was performed back in 1999 and showing the difference. This would be if there was a formula-fed benefit, this would be with a breastfed benefit. And so, with the exception of one study, we see that there's a persistent improvement in IQ scores to about 3 points. And this, again, varies with studies. This effect is much greater in preterm infants. These are all term infants. And there also seems to be a dose response, so that with the longer duration of breastfeeding from 4 to 7 weeks not showing much effect, but more than 28 weeks, you can see a dose-dependent increase in the IQ score. And this was replicated in a recent meta-analysis in 2013 where they even adjusted for maternal IQ, and you can see the forest plot where it's positive and it's also a similar magnitude of effect. And keep this 3-point, 4-point IQ difference in mind as I present some data on milk fat globule membrane. And this is again from that same paper where they looked at the age group, the size of the study, whether the baby is ever breastfed a given number of months. And in most cases, these are all significant, and you can see the degree of the magnitude of improvement. So how might this be working? This is some very nice work from Sharon Donovan, who's at Brown University, and he's been using MRI, and this was from a cross-sectional study, but he now has longitudinal data from the same infants. And so what he's looking at is white matter microstructure, and this study was 133 infants, and they were anywhere between 10 months and 4 years of age. And he characterized them as exclusively breastfed for at least 3 months, exclusively formula-fed, or fed a mixture. And you can see the duration of breastfeeding was around 400 days and exclusive breastfeeding about 150 days. So these were not really long-term exposure to breast milk. And so to orient you, so in the yellow, these are areas of the brain where myelination was greater in breastfed infants than the formula-fed infants. The blue are where breast milk is less than the formula. So when he compared exclusive breast milk with exclusive formula, breastfeeding versus a combination, you can see this persistent improvement, particularly in the frontal region, which is a later maturing region. This was not seen when comparing formula and formula plus breast milk. And then when they found there was a dose effect, so that the infants who were breastfed for a longer period of time had greater myelination. And then when they did some cognitive testing, so this is breastfed versus formula-fed, they found significant improvements in receptive language, and breastfed versus breast plus formula, both receptive language and visual reception. So this is suggesting that when we look at breastfed versus formula-fed infants, there are differences in myelination in the brain. This translates into improvements in cognitive development. So, what could this be? Oops, sorry. So formula is basically milk with vitamins and minerals in it. More recently, we've been adding things like prebiotics. Some formulas have some probiotics as well, but it's quite let's say that's simple, it's not as complex. So as we start to look at breast milk, we have all the essential nutrients, but we also have many immune factors, bioactive components, and the things that could be added to this are the human milk oligosaccharides, which we're learning a lot about now, as well as there's a milk microbiota. And this is something we've just learned about with applying sequencing methods. And the milk microbiota seems to be distinct for women, and it doesn't change much during the course of lactation. So it doesn't seem like it's an artifact. It is composed of some things related to the skin, but it's suggesting that that infant is being delivered not only oligosaccharides, which serve a prebiotic function, but also probiotics. And this is, we're just really scratching the surface and what this might mean. So one of the things that surprised us when we started looking at these human milk oligosaccharides, this is really the fiber of human milk. These are fairly long, most of them fairly long chain. They're not digestible in the upper GI tract. These are just some of the different kinds. You can see sialated and fucosylated and neutral. And the point is they're the third most abundant component in breast milk, so they actually exceed protein. So the question has been, why would nature put all of these oligosaccharides into breast milk in it to a higher level than protein? And they serve multiple functions. So now I'm going to go more deep into some of the human milk components that have been shown to benefit cognition. And a lot of these are from studies where we're adding these components to formula. So we're taking formula, we're adding these components, oftentimes just one ingredient at a time. But LC-PUFAs, milk fat globule membrane, choline, so a number of lipids, gangliosides, but also a protein like lactoferrin has been shown in piglets to improve cognition and, for example, increase BDNF expression in the brain, and the human milk oligosaccharides as well. So just looking at the milk fat globule membrane, I'm only going to kind of focus on one study from this group, but there's a lot of evidence about the importance of milk fat globule membrane. So when milk is secreted by women or cows or really any species, it's actually in a globule. So it's very similar to our lipoproteins in our blood. So this is part of the cell membrane. And embedded in the cell membrane are proteins, glycophospholipids, sphingolipids, cholesterol, etc. And individually, a number of these components in the milk fat globule membrane have been associated with brain function and immune defense. And typically what happens is that in infant formula manufacturing, this is just thrown away. And what we start with is we start with skim milk and we add vegetable oils. And so now there's an interest of, there was a recent editorial, are we throwing the baby out with the bathwater in terms of, maybe we should be using this milk fat globule membrane. So this was a randomized controlled trial from Nicholas Timby and the group with Ole Hernell. And they looked at a bovine milk protein fraction that was added, enriched in milk fat globule membrane, added to formula at 4%. So they had formula fed and breastfed. One of the important components is that the infants on the MFGM formula only received it for the first 6 months. So they didn't actually get the MFGM formula for the first year. And what they found was that, so this is the milk fat globule membrane group, this is the formula group, and this is the breastfed reference. And so when they looked at the cognitive test, there was an improvement in the Bailey score, or the IQ score, of formula versus milk fat globule membrane. And it actually, the levels were similar to the breastfed infant, and this was the first single ingredient ever added to an infant formula that's been shown to basically make up that IQ gap. They also showed in other papers that milk fat globule membrane supplementation reduced infections. The incidence of otitis media, ear infections in the infants getting milk fat globule membrane was very low. And they've only published on the oral microbiota, they haven't published the gut, but they showed that there was a modulation of the oral microbiota and they reduced the abundance of this Moraxella, which has been associated with ear infections, so that's a potential mechanism. In general, because this is a human study, they didn't do MRI or anything. They don't know the mechanism of action. We're actually doing studies in piglets now where we'll be able to look at gene expression in the brain and in the gut. But they're thinking it's the individual or combined effects of these ingredients. There was a previous study in a little bit older children, preschool-age children, showing that adding MFGM reduced fever as well as behavioral scores based on this Achenbach system. So there are several studies out there that showing the inclusion of milk-fat globular membrane is improving IQ in young infants and maybe impacting some of the behavioral assessments in a little bit older kids. So now I'd like to focus pretty much the rest of my talk on the human milk oligosaccharides. So as I mentioned, these are the third most abundant component in human milk. There's been up to 200 different forms that have been identified in human milk samples from women from around the world. It's impacted by the maternal genetics. Most women have about 9 to 10. So as I'll show you, a lot of studies are just adding one, and because not all of them are commercially available yet. But whether one can do what 9 or 10 would do, it's the jury's still out on that. So they're neutral and fucosylated, for example, 2FL. They can be acidic with sialic acid. And some of the data linking HMOs and cognition have been, as I showed you, breastfed infants have a higher IQ. HMOs are 10 to 100 times higher in human versus bovine milk and formula. Fucosylated molecules are present in synaptic junctions and are important in synaptic signaling. And sialylated molecules are detected in the brain. They're higher in breastfed than formula-fed infants, so this was analysis of infants who died from accidents or other non-medical causes, and they found the breastfed infants had more sialic acid in the brain. And we could increase the sialic acid by feeding 3 or 6-SL to piglets. So I'm going to just focus on some of the recent work in our lab, but I wanted to orient you to the fact that people have been studying these HMOs, the 2-fucosyl-lactose, so involved in hippocampal long-term potentiation learning capabilities. This is when they supplemented it to the DAM in the nursing pups. They found improvements in memory and learning. And for 3 and 6-SL, they found diminished stressor-induced anxiety-like behavior. And again, in piglets, increases in the brain as well as modulating the colonic microbiota. So even though these are present in human milk and they're for infants, there is some interest potentially in using them in adults as well. So I want to just finish up talking about a study that we've recently completed. We're still doing analysis. Part of this was presented at the SBGAN meeting. And so this was to evaluate the effectiveness of a combination of bovine milk oligosaccharides and human milk oligosaccharides at improving cognition and altering brain development. We're also doing gut hormone expression and immunohistochemistry as well as microbiome. So that data set is being analyzed. But one of the reasons the piglets are a very good model for the human infant, not only in terms of gut, but also brain. They're large enough that we can do MRI, and we can artificially rear them from birth, which is very, very difficult to do with rodents, which involve pup in a cup type models, which are usually involve a surgery and they're kind of artificial. So we take 2-day-old piglets, and in this study, they receive 2 different human milk oligosaccharides, 2-fucosyl-lactose, or lacto-N-neotetraose, which is also a very predominant HMO that's not fucosylated or sialylated, or a complex bovine milk oligosaccharide, which contain both 3 and 6-SL. So we had HMOs, BMOs, or the combination. So again, the nice thing about the piglet is we've done a variety. This is work in collaboration with my colleague Ryan Dilger. So this is habituation. This is a novel object recognition, which is very translatable to the human infant. We then put them in with 2 different, with actually the same toy, And you can see they're curious like anybody else, and so they investigate this. And they typically will spend about half the time investigating each. We will then delay for either 1 hour or 48 hours, and then we do a test where they now have a new toy. So this is the novel object. And so in this test, we found this is the control group. So again, if they spend an equal amount of time with either toy, it would be 50%. But in the case of the short-term memory, 1 hour, piglets who had been fed the human milk oligosaccharide or the HMO plus the BMO showed improved recognition for the novel object. After 48 hours, we only saw that improvement retained in the infants or the piglets who were getting the combination of the two. So also, Dr. Dilger, because of the size of the pig, we can use standard MRI. We have a specific coil that's developed for the pig, so that's allowing us to get much better resolution. We do a number of measures of brain volume, of microstructure, and metabolites. And what we found in the study that there was no difference in microstructure, DTI, or the metabolites by magnetic resonance spectroscopy. But when we looked at the brain structure, what was interesting is the piglets on getting the HMO diet, so this was a main effect of HMO, so there was no interaction, so we just compared no HMO or HMO. Piglets getting the human milk oligosaccharide had larger volume of corpus callosum in both the left and the right cortex. So this is again suggesting that feeding this in the diet was impacting not only the structure of the brain, but also improving their novel object, their learning and memory, their novel object recognition. We then looked at other regions, and in this case there was an interaction, but there was still sort of a main effect of HMO, both in the lateral ventricle and the pons. So again, we're currently determining what is it about the human milk oligosaccharide versus the bovine milk oligosaccharides, which are more silated versus fucosylated. And so hopefully in the next several months we'll be able to be presenting some of that data. So in this study we found that piglets fed HMOs had larger corpus callosum, cortices, left ventricle, and pons. Piglets fed HMO or HMO plus BMO displayed better novel object recognition memory. However, both were required for a longer delay. We're looking at not only the contents of the genes and the sialic acid in the brain, but also at neuronal branches and things like that. So again, we're proposing that these may be possibly due to altered axonal migration, synaptogenesis, or gliogenesis, and those are things that we're currently investigating. So just to summarize, epidemiological and clinical evidence support improved cognitive development, gut development, and differences in microbiome between breastfed and formula-fed infants. But very few studies have actually started to try to put this together in terms of looking at the gut-brain axis and bringing the microbiome in. And as I showed you, human milk contains multiple bioactive components that individually have been shown to modulate neurocognitive development or the microbiome. And the two examples I gave you were milk fat globule membrane and the milk oligosaccharides. So not only to the study we're doing, but I think in general future research, we need to better understand the mechanism of action of these milk bioactives, and that's we're using models like piglets and rodents are very useful for that. We need to screen the efficacy of isolated bioactive milk components from bovine milk. So many of the components we can add to infant formula are coming from bovine milk, like milk fat globule membrane or sialated oligosaccharides. Some of the human milk oligosaccharides are actually made through chemical synthesis or through microbes. And more of them are available on the market. And actually Abbott just added 2FL to their infant formulas, and Nestle just added 2FL and LNNT, so the same ones that we were studying to infant formulas in Europe. So babies are now getting these formulas. they've been approved, they're grasped, but we still don't really know yet what the short and particularly long-term outcomes are. And so this is this idea of follow-up for longer-term neurodevelopmental outcomes. And, I think it's important also to start to look at other neurodevelopmental outcomes such as ADHD and ASD. And I know the group in Cork and others are starting to look at this because the data is not as clear. The epidemiological data in many cases show that breastfeeding protects or reduces the incidence of ADHD and ASD, but it's confounded by whether the mother also has ADHD, and obviously there's a genetic component. But I think that this is a rich area to investigate. So I'd just like to acknowledge all the people who do all the work, Dr. Monaco and Dr. Wong in my group. This is Dr. Dilger, who does all of the neurocognition. I do the gut and the microbiome. And he has two excellent students, Stephen, who does the behavioral testing, and Austin, who does the neuroimaging. Some of our grant support, and this is actually a figure from we had a paper published in Frontiers in Young Minds. It's called What's in Milk? And so I just think it's a very cute figure. So thank you.